Hello ladies and gentlemen, welcome back to the lymphatic system and immunity. We have been going through talking about how the different parts of the immune system work, and last time we talked about the different processes. Today we are going to continue with the third part of immunity, and we are going to talk about the function of the different types of antibodies. By the way, antibodies are also known as immunoglobulins. If you see an abbreviation like these with the I, little g, and then a letter after it, that's all it means, is these are different types of antibodies. You do not, by any means, need to know these. I just wanted you to realize there are different kinds. But you can see that same basic, kind of almost a Y-shaped structure for the antibodies. What we're going to talk about today is how these antibodies actually work. We know that the plasma cells make them, and we know that the antibodies fight disease, but we haven't talked about exactly what they do. Okay, the first process we're going to talk about is called neutralization. And neutralizing something basically in this sense means to make it harmless, something that cannot damage. Okay, so what you can see is on viruses or bacteria, basically what happens is these Y-shaped antibodies just cling to them. And what happens is it prevents the bacteria or the viruses from actually being able to touch your cells, and it prevents them from being able to damage your cells at all. You can also see on this right side right here, they can also bond to toxins that are released by the bacteria, preventing them from entering in your cells. So of course, if these things can't enter into your cells, they can't damage them. So that is neutralization, to take something harmful and make it harmless. Okay, the second process is called precipitation. Now, precipitation, we usually think about rain and it falling down, or snow falling down, ice falling down from the sky. Same idea here. Okay, you have basically these antigens, the disease-causing whatevers, these little orange thingies, and you have the antibodies, these Y-shaped things, and what happens is they start to clump together. And what happens is they form these large complexes called a precipitate, which is basically just thinking pretty much like a solid almost. And because they are clumping into these large structures, they actually will settle to the bottom of the fluid they're in or whatever, and they will be unable to function. They aren't going to move around. It's kind of the same idea as a sticky trap for a rodent. They get stuck. They can't go anywhere or do anything. Okay, so that is precipitation. They get heavy because they stick together and they fall to the bottom so that they can't go anywhere and cause harm. The next process is one you've heard of before, and that is agglutination, and you can see it with the red blood cells that are here. Okay, you have the red blood cells. They have their antigens on the surface. If you have the type of antibody that will fight against that type of antigen, what happens is they all clump together. Now, the difference between this and precipitation is that by clumping together here, it actually makes it more efficient for phagocytosis to happen, which, remember, is those macrophages, those big white blood cells coming to eat and destroy the cells. Okay, so by having agglutination, whether we're talking about the wrong type of red blood cell, or whether you're talking about bacteria or a virus, all of them clump up, and that allows the macrophages to eat them more easily. Okay? The next one is called opsonization. Isn't that a beautiful word? This is similar to agglutination in that it improves phagocytosis, but this is where the antibodies actually will attach to the pathogen, to the disease causer, whether we're talking about bacteria or viruses, protists, whatever. The antibodies attach to it, and that makes it unable to go anywhere or travel anything, and it actually helps the macrophage attach to it better so that phagocytosis can happen more easily. It allows them to stick in place to that white blood cell so they get eaten quickly. Okay, the next type is immobilization. Immobilization is basically where you have these bacteria or protists, and they have these flagella, which you should remember back from talking about cell structures long ago. In the process of immobilization, what happens is the antibodies will attack and cling to or destroy the cilia and flagella that allow these bacteria and protists and little microbes to be able to move. And it just stops them. And they can't go anywhere. They just end up floating through. Now, this technique does not work on viruses because viruses have no ability to move themselves. They only move where your blood or something takes them. But this would happen against protists, like amoebas. It would happen against bacteria. This would be an example of bacteria, so it would stop their motion. Okay, the next process should be the one you're most familiar with because it is part of the process we've talked about in the immune system. And that is where you actually have the antibodies attach to a cell, whether it's bacteria or whatever, and it actually will attract the white blood cells to it. So, we've talked about that a little bit with bacteria. They release these substances, and that attracts them. In this case, you have the bacteria or whatever, and these antibodies attach themselves to it, and wherever it travels, it's attracting these macrophages that are going to come and eat it. And it'll also help with the B cells and the T cells and all those different processes. And that's just basically inflammation, which you know of as swelling. When you get an infection and it gets red and it gets swollen and it gets hot, that is the process that's happening, is chemicals are being given off because you've had bacteria or whatever enter that cut or whatever it is, and it swells up so that all those all the white blood cells can come in and destroy the bacteria that are there. It's red because the blood vessels are open. You get pus because those are white blood cells that end up dying if they get overwhelmed by the infection. Okay, so it's swollen because the blood vessels are opening up. It's red because the blood vessels are opening up. And it is hot because the blood vessels are opening up. And all of that is the part of the process of inflammation and functioning of your immune system. Okay, if you have questions, make sure you ask. And this is about how the antibodies do their thing. Until next time.